my name's Abigail Dunham, and the subject that I chose to research was cloning. So first, I'm going to tell you about some of the big scientific cloning things that they have done. So first, I'll talk about Dolly the sheep. Dolly the sheep was born July 5th, 1996, in a Rosen Institute in Edinburgh, Scotland. She was revealed to the public on February 3rd, 1997. So as you can see, they didn't release her to the public for close to a year. She was the world's first mammal to be cloned from an adult cell, and she was considered the most significant scientific breakthrough ever. She proved that adult cells can be reprogrammed themselves into new beings. Even though the scientists try to make her cells young again, they think that they're older because she had a lung disease that was not typical in young sheep, and she also had arthritis even though she was only 6 years old. She bred on 2 different occasions with a Welsh ram named David, and over her life gave birth to 4 little lambs. Francis Crick and James Watson are the first pioneers in cloning technology. The next one is Snoopy the dog. Snoopy is the first dog to be successful. He was produced by a team of South Korean researchers from a single cell from an Afghan hound's ear. Dogs are very difficult to duplicate, so this was a huge deal. Mostly because their eggs are not as easy to extract as eggs are from other animals like cows or pigs. Thanks to Woo-Suk Wang, he succeeded where others had failed. In 2008, Snoopy fathered puppies of his own, and there was no cloning required. Okay, so next I'm going to talk about somatic cell nuclear transfer. This is a laboratory technique used for creating ovum in a donor's nucleus. It can be used in embryonic research or in regenerative medicine, where it can be referred to as therapeutic cloning. It can also be used in the first steps in the process of reproductive cloning. This is where the nucleus is removed and the rest of the cell is discarded. At the same time, while the nucleus of an egg cell is removed, it is then inserted into the eunucleatic egg cell. After that, it is reprogrammed by the host cell. The egg that now contains the nucleus of the somatic cell is stimulated with a shock, and then it will begin to divide. After division, the single cell forms a blastocyst, which is the early stage embryo with about 100 cells with almost identical DNA to the original organism. Hwang Woo-Suk is a geneticist in South Korea. In 2004 and 2005, he was claimed to be one of the first people to have cloned a human embryo for the first time, and that he had derived that from 11 stem cells. He was a professor at Seoul National University in South Korea. He stated that all the women that donated their eggs to his laboratory were volunteers and did not receive any compensation in return. In 2006, he admitted that many of his results were fabricated. An investigation was done and found that more eggs than they had accounted for in their experiments had actually been people who were paid for their eggs. This sparked lots of debates around the ethics of egg donation for research. The reason that suspicions had occurred is because one of the female researchers, named Jamin Koo, she was a co-author in the 2004 publication, and admitted that she had donated oocytes to their research. When confronted with all of this, Wayne denied all allegations of ethical misconduct and said that the accusations occurred from a misunderstanding because of her lack of English. He lost a U.S. co-author because of this. In 2006, they found that 2 research scientists had donated eggs that many donors had been paid to donate. So investigators found that Wang had collected over 2,000 oocytes from at least 119 different women, and 2 were from his laboratory. 85 of these women were either paid or offered extra benefits. These were in violation with the 2001 Korean Guidelines of Medical Ethics and the 2005 Bioethics and Biosafety Act. Some consider this to be unethical to pay oocytes donors because it can be a form of pressure. He resigned from his position as professor in 2005. Research was done and came to the conclusion that these women were under societal or personal or work-related pressure to donate their eggs and were not fully informed of the details involving in the egg retrieval procedure. Taking oocytes from women involves physical and psychological risks. Because of these risks, research participants are supposed to be informed of those risks, and they are not properly informed. When the eggs were being collected, there was no informed consent, and they only had informal consent. In some cases, women's ovaries were actually removed completely out of their bodies with no consent. Numerous suffered from ovarian hyperstimulation, and some had to be hospitalized after their oocyte extractions. Wang also used illegal egg brokers to pay women. He did this because women who usually undergo fertility treatments received discounts for reproductive treatments in exchange for their eggs. Paid women were normally in their mid-20s and significantly younger than volunteer donors who were in their mid-30s. Some of the reasons that he was able to collect so many eggs unethically is because Korean couples who had fertility issues and were doing in vitro fertilization often purchased their eggs on the internet, and he used these egg brokers to get the eggs. It actually showed that 2 of his researchers had donated their eggs, and he made 8 other of the people working on this sign a contract saying that they would donate their eggs if they needed to. These women who donated their eggs did it out of national pride, and they wanted to help others. In 2009, Wang was convicted of embezzling money and bioethic violations. He was denied his license to continue research on human cloning, but later researched cloning animals. He started an organization called the Soam Biotech Research Foundation in Seoul, and they created the first clone dog in 2005. So looking at some of the recent developments in cloning technology, they can be used for stem cell research. Stem cell research is actually working on helping create cures for Alzheimer's and Parkinson's disease. It is also working on bone marrow, spinal cord, cellular. But a lot of these things are usually demolished because of the ethics that go behind when you're having to get them. The idea of cloning a deceased human or pet has fallen out of favor, which you can all assume why it has fallen out of favor because it's a little weird. In 2008, there's no desirable difference between cloned and non-cloned cows, so goats and pigs. It is allowed on all of them, mostly for breeding purposes, not so much for the meat production. One of the biggest breakthroughs is the ability to turn a genetically modified cell into a fully developed animal, which is awesome. So the questions are, what are medical benefits and dangers of animals in human cloning? So some medical benefits are that they can be used for stem cell research in different conditions, such as Alzheimer's or Parkinson's disease. They can also be used in stem cell research for bone marrow, spinal cord, and cellular. Some dangers are that if they do it unethically, like Wang did, and cause a bunch of other medical issues, or psychological problems as well. So that's I would consider that a danger. Some medical benefits for animals are that they can genetically modify animals so that their cells and organs can be transplanted into humans. The next question is, what form of cloning are legal in the U.S. and what about other countries? So animal and stem cell cloning are legal in the U.S. In 46 countries, human productive cloning has been banned. France, Germany, and Canada banned the creation of cloning embryos, and the UK authorized creation of cloning embryos for research. When looking at the question, what moral arguments for and against animal and human cloning, some of the moral arguments are that they can be really helpful for medical things. Like I said before, it can be really helpful for looking at advances in Alzheimer's and Parkinson's disease. That's super awesome. And for animals, if you can clone an organ from an animal to be put in a human, that's so awesome. But if you look at the way that they have been going about doing these things, it's very unethical and they do not go about it correctly. Well, that's the end of my presentation. I hope you guys learned a little bit more about cloning and the cool things they have behind it, but also some of the things that aren't very ethical, like Wang, who did all that stuff to those women. That was so unethical, and they definitely should have been informed. I'm sure that stuff like that is still happening today, and that's definitely not okay. but there are also some great advances in cloning. So I hope you learned a little bit more about it and enjoy this presentation. Thanks.